Welcome to our channel Bio by Gulab Sir for Wisdom Coaching Center. As a part of series of lectures, today's topic is glycolysis. As we know that every organism need to be respired. Respiration is the process of consumption of organic matter to produce the energy which is required for all metabolic reactions going on in cell, tissue, organ system and organism. Even unicellular organism also need to be respired. Either it is aerobic or anaerobic, but they respire with in presence of or absence of oxygen. Whether they are prokaryotic or eukaryotic, but still they need to be broken down organic material into simple organic or inorganic substances to yield the energy. So as the today's topic is that respiration, the first process which involve as a major part of that respiration is glycolysis. So glycolysis is a stepwise breakdown of glucose into pyruvic acid. You are aware about that organic substances are the substrate for respiration, whether it is glucose, fat, protein or organic acid. All they undergo breakdown, but in presence of glucose, fat remains stored. Protein comes after fat if there is a long fasting that organism is not supposed to getting carbohydrate and protein, then they do respire with the help of fat. So glycolysis is a stepwise breakdown of glucose into pyruvic acid. So glucose. So first step of glucose is phosphorylation. As we know that glycolysis is stepwise breakdown of glucose into pyruvic acid. It is also known as Embeden-Mehrob Paranas pathway. This has to be remembered as another name for glycolysis, since it is discovered by Embeden, Mehrob and Paranas. These are the three different scientists who discover and explain glycolysis. So glucose is a stepwise breakdown of glycolysis is a stepwise breakdown of glucose into pyruvic acid. First step which involved in this is phosphorylation. Where glucose undergo phosphorylation with the help of adenosine triphosphate, adding one phosphate in glucose, producing glucose 6-phosphate. So this is byproduct of that first process, phosphorylation. Where this process is going on? Cytoplasm in eukaryotic cell. Cytoplasm of eukaryotic cell. Prokaryotic as such, they don't have nuclear membrane. Therefore, it is protoplasm. So in prokaryotic cell, it goes in protoplast. And in eukaryotic cell, since protoplasm is divided into two parts, like cytoplasm and nucleoplasm, so it goes in cytoplasm of eukaryotic cell and protoplast of prokaryotic cell. It can occur in presence of oxygen as well as in absence of oxygen. Therefore, it can occur in anaerobically respiring organism as well as aerobically respiring organism. So first step we learn, that is phosphorylation, where phosphate is added into glucose to form glucose 6-phosphate. It's very easy. When we say that phosphorylation, what does it mean? Addition of phosphate into glucose and producing glucose 6-phosphate, 6 carbon, as glucose is hexose sugar having 6 carbon in its composition. So this phosphate from adenosine triphosphate release and attach with glucose to form glucose 6-phosphate, phosphate attached to the 6 carbon of the glucose in their chain. Now this glucose 6-phosphate undergo isomerization. So second step is isomerization. That means formation of isomer of glucose. What is isomer of glucose? Fructose is the isomer of glucose, where rearrangement of that molecule occur to form its isomer, fructose 6-phosphate. So fructose is also a hexose sugar. Only rearrangement of hydroxyl and hydrogen ion made it different than that of glucose. Otherwise, chemical formula is same, only physical formula different, structural formula differs, and therefore it is an isomer of glucose. Here, this occur in presence of hexokinase enzyme and magnesium ion. This occur in glucose phosphate or hexose phosphate isomerase. Hexose phosphate isomerase, as well as a cofactor magnesium. Now this fructose 6 phosphate again undergo phosphorylation, that is second phosphorylation, as first one occur here, second one here. Again phosphate from adenosine triphosphate release, convert into ADP and form fructose 1,6 diphosphate. Fructose 1,6 diphosphate. Here you can see that another phosphate will release from ATP combined with first carbon of the fructose, and therefore already it was having one more phosphate with 6, therefore it becomes 1,6 diphosphate or biphosphate. Now this fructose 1,6 diphosphate undergo cleavage. So next step is, fourth step is cleavage. That is called breakdown of the 6 carbon compound into 2 3 carbon compound. One is dihydroxyacetone phosphate and another is phosphoglyceraldehyde, that is triose phosphate glycerate. Phosphoglyceraldehyde also known as triose phosphate glycerate. They are interconvertible. They are interconvertible. They can be converted into each other, and that enzyme which catalyze is phosphate triose isomerase. Phosphate triose isomerase. So they are interconvertible. Both are 3 carbon compound. Hence, this 6 carbon compound split into 3 carbon compound, one molecule of DHAP and one molecule of PGL. Now, this one molecule of PGL, that is not sufficient to produce 6 carbon compound C6H2 to produce 2 molecule of pyruvic acid. Therefore, this interconvert DHAP convert into PGL, and therefore 2 PGL undergo this process called dehydrogenation. So it happen in presence of PGL dehydrogenase and that NAD convert into NADH2. That what NAD is to as a byproduct, as well as there is addition of phosphate from H2PO4, that is phosphorylation. So here PGL undergo oxidation as well as phosphorylation. So here this is not substrate-based phosphorylation. Okay. So here two molecules of 1,3-bi-PGA, biphosphoglycerate or biphosphoglyceric acid is being produced. Now this happen in presence of enzyme PGAL dehydrogenase and produce 1,3-biphosphoglyceric acid. Next step, the 2 molecule of 1,3-biphosphoglyceric acid undergo dephosphorylation, where 2 molecule of ATP are produced from 2 molecule of ADP, because 2 phosphates are released from each one. There are 2 molecule of each one and form 2 molecules of 3-PGA. Now further this 2 molecule of 3-PGA, 2 molecules of 3-PGA undergo again, that is called isomerisation, produces 2 molecules of 2-PGA, that is what phosphate, which was attached at 3rd carbon, now attached at 2nd carbon of the phosphoglyceric acid, and naturally this state is known as isomerization. Here this was fifth stage, dehydrogenation. Now this is isomerization. This step is sixth, isomerization. So here this undergo isomer, that is phosphoglycerate isomerase in presence of PGA isomerase. That enzyme is very easy to remember. Process is isomerization, substrate is phosphoglycerate, so phosphoglycerate isomerase, and it's very easy to remember also. Now this dephosphorylation which was occur in presence of enzyme kinase and cofactor magnesium. We have to remember this cofactor as and when they are being used to convert. Here also dephosphorylation occur in presence of fructose or hexose phosphate, fructose phosphate kinase, and that cofactor is magnesium. So see, at these three places in continuation magnesium act as a cofactor. This again kinase, as and when there is a phosphorylation or dephosphorylation, that process occur in presence of kinase enzyme is very easy to understand, and that too cofactor along with that kinase is always magnesium. Okay, here again 2-PGA undergo dehydration. So next step 7, that is dehydration, that is loss of water from 2 molecules of phosphoglyceric acid into 2 molecules of phosphoenol pyruvate. So as here this what, because of release of water it happened, therefore the process is dehydration. It occur in presence of enzyme enolase. Now the phosphoenol pyruvic acid undergo next step, last step is dephosphorylation, that is 8th stage. Phosphoenol pyruvate convert into pyruvate, dephosphorylation. Again 2 molecule of ADP producing 2 molecule of ATP. Finally that phosphoenol pyruvate convert into 2 molecule of pyruvate. Thus here this process is completed and 2 molecule of pyruvic acids are being produced. Again this is called catalyzed by the enzyme called phosphoenol pyruvate kinase, and it happened in presence of magnesium as well as K positive. So these are the 2 factors here, and that is the point of NEET. So question can be asked in NEET. So to remember that minerals which catalyze last process of glycolysis to convert phosphoenol pyruvate into pyruvate is catalyzed by phosphoenol pyruvate kinase in presence of factors A, B, C, D. These options will be given. Now see how many process, 1, 2, 3, 4. This dehydration is 5th, and this dephosphorylation is 6th. This was again dephosphorylation was the 6th process. So continue in the number that 7th, 8th and 9th process. So here to have the correct explanation with that sequence of number, here once again as a correction, 6th, this is the 7th, this is the 8th, and this one is the 9th. So total 9 steps involved in the process of formation of pyruvate through glycolysis. So finally that glucose is converted into pyruvic acid. Now pyruvic acid can enter into the Krebs cycle to have the complete breakdown into water and carbon dioxide, or undergo incomplete breakdown to form the either lactic acid in our muscle cells. When we run fastly or vigorous exercise, then there is a lack of oxygen. So in our striated muscle also that anaerobic respiration can occur, and therefore sometimes we feel cramp. as well as anaerobic respiration carried out by microbes, and that process is known as fermentation. So byproduct is alcohol. So pyruvic acid further undergo anaerobic breakdown or aerobic breakdown. If it undergo anaerobic breakdown, byproduct are alcohol and lactic acid, and aerobic breakdown, that is what Krebs cycle, which occur in mitochondria inside the eukaryotic cell, will have complete breakdown of pyruvic acid into water and carbon dioxide to yield full energy, and therefore 36 ATP is the net product of aerobically respiring organism during respiration. But here, as a part of first step of respiration, that is glycolysis, stepwise breakdown of glucose into pyruvic acid, that we did understand. Point to be note down, that is in board examination, question can be asked, draw a schematic flow chart of this glycolysis. This is a schematic flow chart. Question can be asked, what is EMP pathway? Explain it with schematic flow chart. So in addition to this schematic flow chart, explanation include this 9 step, and it's very easy to explain. See, for example, if we have to explain phosphorylation as a part, glucose undergo phosphorylation producing glucose 6-phosphate in presence of ATP and hexokinase enzyme and magnesium. जो दिखता है वही लिखना है. So glucose 6-phosphate undergo isomerization producing fructose 6-phosphate in presence of hexose phosphate isomerase and magnesium. Third step, underline it and write it. So you can have underline to the major steps. So phosphorylation, photophosphorylation, again phosphorylation, where fructose 6-phosphate undergo phosphorylation in presence of ATP, fructose and fructose phosphate kinase to produce fructose 6-biphosphate. Like this, every step we have to explain. So you can underline this major step as a part of highlighting the steps. Second, you also please remember that magnesium, which steps are carried out by magnesium? One of the following steps is not catalyzed by magnesium as a cofactor. So question can be asked in NEET. These are the steps which are catalyzed by magnesium. So in that option, they may give one of the step which is not catalyzed by magnesium. Therefore, question can be asked that one of the following step during glycolysis is not catalyzed by the cofactor magnesium. Like this also question can be asked, find odd one in glycolysis, that these steps are catalyzed by cofactor magnesium along with its enzyme, and one of the step is not catalyzed, that will be the odd one. Like this. So question can be asked, if one glucose molecule undergo breakdown, so how many number of ATP will be produced during aerobic and anaerobic reaction? That we have to calculate. So here to understand, if anaerobically respiration occur, so total number of ATP are produced anaerobically, 2 plus 2, 4 ATPs are produced over here. This 2 ATP plus this 2 ATP, total 4 ATP produced. And 2 ATP are utilized, 1 and 1. Here this one is utilized, this one is utilized. So net production of ATP in anaerobic reaction, if this glycolysis occur anaerobically, then the total number of ATP are produced, we can see on next page. So I take next page to understand. As a part of glycolysis, now to understand total number of ATP produced, net ATP produced during aerobic and anaerobic respiration. If that breakdown of glycolysis, glucose, is occur anaerobically and aerobically, how many net ATP are produced? To understand, we have to make the balance sheet. Total number of ATP produced during anaerobically. To understand, once again we can refer the previous page. Here total number of ATP produced during anaerobically, 2 ATP plus here 2 ATP. So total number of ATP produced are 4. So here 4 ATP. Now total number of ATP used, total number of ATP used are 2 ATP. Where they are used? for phosphorylation. To understand, we can come here. Here at two places phosphorylation occur, 1 and 2. 1 and 2. So 2 ATPs are used. So we can mention there 2 ATPs are used. Therefore, net production of ATP. Therefore, net production of ATP during anaerobically respiring or anaerobically respiration or anaerobic respiration is 2 ATP, and therefore whenever we do anaerobic reactions, one that lactic acid formation, so C6H12O6 anaerobic will break into 2 molecule of lactic acid producing 2 ATP, and these are the 2 ATP. If it is fermentation carried out by microbes, that is either yeast or bacteria fermentation, so alcohol is being produced, so 2 molecule of C2H5OH plus 2 CO2 plus 2 ATP. Here also 2 ATPs are produced. So how these 2 ATPs are produced is explained here, because both the reactions are anaerobically occurring reactions. Since lactic acid producing on muscles or fermentation carried out by microbes producing lactic acid and alcohol with the product of 2 ATP, and this is what explanation for it. This total number of ATPs are being produced. Now aerobically, how? Total number of ATP produced 10 ATP. To understand, once again we can take the reference of previous explanation here. These 2 ATP produce here, 2 ATP is produced here, and this NADH2 during aerobic reaction, this NADH2 is transferred into mitochondria where it oxidizes, and when one NADH2 oxidize produce 3 ATP, so 2 number of NADH2 oxidize producing 6 ATP. So these 6 ATP which are produced by the oxidation of NADH2 in aerobic respiration are in addition to these 2 ATP and these 2 ATP during dephosphorylation, 4 ATP plus 6 ATP, 10 ATP. Hence, we have written here 10 ATP. I hope you did understand how total number of 10 ATP produced if glycolysis occur aerobically. Now, as we already understood that 2 ATPs are consumed for phosphorylation during these 2 phases. This is one phosphorylation and this is one of another phosphorylation what's shown here. So here 2 ATPs are used, so we can write here minus 2 ATPs which are used for phosphorylation. Now net production, we have already written here net production of ATP. So in front of it, net production is 8 ATP. So total number of ATPs produced during aerobic respiration in glycolysis are 8 ATP. So here we did understand balance sheet of ATP production during aerobic and anaerobic respiration. So here we have completed the topic glycolysis as a first part of respiration process. I hope you did understand. Even if you are in problem, you can put into the comment section, and I appeal you to subscribe and like our channel, as well as press bell icon so as to receive videos in a series time to time as and when they are uploaded. Thank you.